Welcome. This is a nice problem, not having enough seats. That means that a lot of you are here for our first seminar of the fall. My name is, good evening, my name is Adelisa Flores. I teach chemistry here. And I want to thank you all for coming. We have a very special seminar tonight. I'm part of the seminar planning team on the LPC side, and I've been doing this for maybe 7 or 8 years already. Just a brief history. This seminar series, we have 4 seminars every year. This was originally conceived by our now Dean Nano. You see her walking around here when she was a biology faculty here. So we have her to thank for getting this seminar series started. Real quickly, just a few acknowledgments here from the lab. We want to thank the speakers, of course, for accepting our invitations to come to the campus so that they can talk about their work. And also we want to thank Joanna Albala. I saw her. Where is she? She's right there. She's from the lab. She's the Education Outreach Manager of the Science and Technology Education Program. She and Chris Culp. Chris Culp is the group leader in the Biosciences and Biotech Division. Both of them have been really, really helpful in getting us these wonderful speakers come to the college. From the LPC side, I want to thank, there's lots of faculty I saw earlier coming in. Thank you for coming, and thank you for bringing your students or encouraging your students to come to the seminar series. I want to thank classified people as well, and I know some administrators have attended these as well. We have our dean, of course, Dean Nano over there. The dean of the STEM division. Let me see, anyone else? Okay, all right. From the planning team, LPC, I want to thank Carol Schill, and I think she walked out for, oh, she's right there. I want to thank her. She's really the person who kind of keeps the gear grinding to get us to this point, inviting and helping with the speakers and helping get them prepared for tonight. I want to thank David Lee and Christine Hornbaker, IT people, helping us set up all these nice AV stuff here. And I want to thank the Dean's office, Linda Cross in particular. She's been very helpful as well for all the logistics. A couple more people, Giselle Nunez at the district for helping publicize the seminar, and then Tim Drulli, our webmaster, who helps us maintain the website for the seminar series. And then a few more people here. I have in the planning team, I have Mike Ansel from chemistry helping out, and we have Robin Rehagen. If you ever wonder who makes those stunning flyers and posters for the seminar, that would be your physics teacher, Dr. Robin Rehagen. She's been working on those. And Barbara Zing, I saw her. She's also part of the planning team. I know she came in here. And then Keith Level from engineering. I want to acknowledge Dr. Neil Eli. He was our former dean. He was the dean when this seminar series started, so he's right over there. And also Dr. Lisa Everett, another former dean who was involved in the planning. And lastly, Dr. Beth Vitalis and Dick Farnsworth from the lab, who were part of the original team that brought this seminar series to the campus. Now, finally, on to our seminar for tonight, flexible microelectrode arrays for monitoring brain activities. So our speakers are Dr. Anna Marie Belle and Dr. Allison Yorita. They're from the Center for Micro and Nanotechnology and Bioengineering Center Materials Engineering Division at the lab. So just a few tidbits about them. Dr. Annabelle is a research engineer. She's also a principal investigator at the same division. Her work focuses on developing biosensor arrays for research into neurological diseases and disorders. Her PhD is in analytical chemistry and she got it from the University of North Carolina at Chapel Hill. And she has also established the facility for in vivo testing of neurological biosensors at the lab, and is a member of the Kavli Institute for Fundamental Neuroscience. Dr. Yorita is a postdoctoral researcher at the same division, and her PhD is in chemical and biomolecular engineering from UCLA, and where she worked on microfabricated devices to detect neurotransmitters and nucleic acids. Her work at the lab now focuses on microfabrication of neural devices, as well as studying and characterizing chemical sensing capabilities on flexible polymer probes. I'm sure you will hear a lot more about their work, so without further ado, let's give them a big round of applause.
Thank you very much for the introduction. Can everyone hear me okay? Perfect. So, as you might have guessed from the title of this talk, I'm going to be discussing with you some of the flexible microelectrode arrays that we make at Livermore. I'm assuming being in Livermore, you guys know where Lawrence Livermore National Lab is, or you have heard of it at some point in your lives, correct? Yes. What a lot of people don't know, though, is the lab actually has a lot of work going on in bioengineering. So again, starting even as early as the 70s, there was work before that as well, work looking at ways to see what cells are doing, what's happening in cells. There's been all kinds of new cell sorters, fancy technologies to figure out, again, what is going on in the body. And in recent years, starting in the 2000s, that work went not just into building diagnostic platforms, but actually building new ways to test what's going on in the body and help with human health. Right now, the center has evolved to have kind of three main areas. We have what we call ex vivo systems. So that's, if you know about bioprinting or organ on a chip, you always see these kind of news articles coming up about people making tiny brains that they're testing new pharmacological agents in, things like that. That's kind of that center there. What we're doing is a lot of bioprinting efforts to try and come up with better therapies to help with human health. We also have the in vivo systems. I'm going to just quickly say that's most of what today's talk is about. I just wanted you guys to know that there's a broader breadth. And then for both of the in vivo and ex vivo work that we're doing, what's happening there is there's a huge computational component. There's predictive biology to figure out what are the best cells to use in these systems, but also the arrays that Allison will talk to you guys about in a little bit. We get so much data off of those, and then the problem becomes, what do you do with so much data? It would take a human their entire lives to go through it. We need computers to help us figure out what these signals from the brain mean. So this is all a huge part of this center. But today we're going to be mainly focusing on these in vivo systems and the microelectrode arrays that we actually build to go into the brain. Again, so a neural interface or microelectrode array in the brain, this is probably what you guys think about from the movies, right? This is kind of a big room, but I'm going to give like 5 seconds. If you guys know what any of these are, feel free to yell them out. Right? What about the bottom on the right? Right? So this is kind of what Hollywood or what people think about when they think of neural interfaces, right? These things that go to your brain, that let you run robots, and like everybody has them just for better living. And what this really means, though, is any device that interfaces with your peripheral or central nervous system. So your central nervous system is your brain. Peripheral nervous system are all of the nerves kind of throughout your hands and arms, your spinal cord, this whole system that allows the rest of your body to so rapidly communicate with your brain. And so anything that interfaces to either of those devices, or either your central or peripheral nervous system, can in fact be considered one of these neural interfaces. The reason it's called a neural interface is it interfaces to neurons. Neurons are brain cells. This right up here is a brain cell. What it is, there's a cell body. The way that cells can communicate so quickly in your brain and throughout the rest of your body is because they use electrical signals. And so these electrical signals move down this axon terminal that you can see, and they move there more rapidly than chemical transmission would happen, right, because of the rapid electrical signals. So what we can kind of do is the same way that the cells themselves use these electrical signals, we can also kind of tap in and listen into these signals, hear the electrical signals, and then also send electrical signals into the tissue so they can hear what's going on. So real-world neural interfaces aren't quite as cool as the things you see in Pacific Rim or Star Wars, but maybe we'll get there. Right now, some of the examples that you guys might be familiar with are cochlear implants. Have anybody heard of cochlear implants? Right, so that's a standard one. That's a neural interface. It's a way to allow an instrument to help directly interface with your brain. Deep brain stimulation is another one that's used for a lot of different therapies, everything from Parkinson's, pain management, and a bunch of different disorders. And then the one that I'm going to talk to you about today as an example of a neural interface is called the artificial retinal. And that's actually an implant that goes against the eye to help with vision. So the reason we're using this as an example is this was a huge project that happened across many national labs a few years ago to help create vision for those who do not have it using a neural interface. So what this is it is an electrode array that gets put on the back of the eye. What then sees something from a camera, what's on that electrode array, is then stimulated on the back of the eye in a pattern. And so essentially it creates vision for someone. This was a huge advancement. We won a lot of awards for it as a lab. It's also really cool. I'm going to show you some videos in a second. Like, it actually works, right? Sometimes things seem really cool, but if it doesn't help people, it really doesn't matter maybe quite as much. So normal vision, how you see normally. Typically what will happen is some light or signal comes in, hits your retina. Then what happens is that signal is moved forward through all of the rod and cone and other cells in the back of your eye, through the optic nerve back to your visual cortex. And that's where your brain then has this visual signal that you see, you interpret what you're seeing, vision. What happens though, normal vision, this is what's happening, right? The light's coming in, you're getting this picture, it's hitting the retina, that signal is now moving forward to your brain. Unfortunately, what happens in retinitis pigmentosa and then macular degeneration is you start to lose vision. So in this disorder, what happens is you start, it kind of looks like you're looking through a toilet paper tube, and then a paper towel tube, then a wrapping paper tube, and then nothing, just your vision kind of closes in. And then macular degeneration is the opposite, where you start with a dot and work out. And in both of these disorders, what happens is the rod and cone cells in the retina kind of break down and can no longer send that signal forward to, through the optic nerve to your brain. So what the artificial retina does is exactly what it sounds like. It acts like an artificial retina to directly stimulate and send signals through the optic nerve to your brain. Again, it is exactly what it looks like here. A person would wear a camera on glasses. The camera could be in theory anywhere, but this is easy for users. It's something they're used to naturally moving their heads. That signal is taken in. It is then transmitted and turned into electrical signals by this nice little purse. That is then used to stimulate a pattern, in this case, the E from the eye chart, on the back of the eye. And so, again, I want you guys to see some videos of people using these, both with and without. This is going to be somebody walking a line. So this first time, she is going to walk with the implant off. And again, it's a relatively large line, but she is not able to see it. I feel like I need to speed it up a little. You probably got the idea already, didn't you? And so here, the implant has now been turned on. And so the idea, again, is with this, you can have someone who now can see where doors are, can move around a little better. This next video, what I want you guys to look at is on the bottom right. As the video starts, this woman is wearing the implant. She's going to be tracing letters and saying what the letters are. You will hear that she gets them all right, which is great. But the thing that's cool to watch is in the bottom right, you're going to see what she's seeing with the camera and which electrodes on the back of her eye are stimulated. So kind of use that as a way to see if you think you could guess, based on getting how gradiated the signal is, information. So again, you can see in the bottom right, when the dots become white, that's the stimulating electrodes that are happening. Again, I don't think you can hear it. It sounds kind of quiet, but she's getting all the answers right. And what I find impressive is this is a C, and this O, I'm not sure, they don't look any different to me on the pattern. But this is a good example, I think, of how the body can start to learn what these signals are. A huge part of neural interfaces, and one of the things we'll talk about later is how much we're learning from creating these machines that interact with humans. The responses we see aren't always what we would expect. There are people who talk about seeing colors with these, but again, it's just an on or off signal. Again, this was the world's first thin film prosthetic. Thin film because it was microfabricated instead of handmade, which is the way a lot of electrodes up until this point were made. This particular example here is 240 electrodes. The one that was being used by patients that you just saw in the earlier videos had 60 electrodes. And so we did work not just on this electrode array, but the packaging, right? So there's tiny electronics that go in. There's all sorts of systems integration. But one of the big things that the facility we work in and the national lab work on is making these higher density arrays using microfabrication. And so Allison in a second is going to show you how that's done, but I just want to give you an example of why we do it. So can any of you guess what this is? Now?
George Washington. Over next time.
Have you seen this before? You're an excellent candidate for a 16 electrode array. So again, it gets a lot easier, right? So imagine each of these is a doubling of the pixel size, right? Or essentially doubling the number of electrodes. So this is a good example of, in the case of vision, why having more electrodes and better resolution would be very helpful, right? But you can imagine in getting information from the brain, that same kind of thing. The denser we can get electrodes, the smaller we can make them, the more information we can get, the more targeted therapies can become. And so with that, Allison will talk to you about how we do this patterning.
Thanks, Anna. Can everyone hear me okay? Perfect. So next I'll be talking about, as Anna mentioned before, we need to go to higher and higher densities, more and more of these electrodes packed into a tiny space in order so you can see, Albert Einstein better, for example. So what kind of technologies do we actually use to get this to work? So this is all done via microfabrication. So you might notice we have what's called a clean room at the lab. You see everyone wearing these kind of bunny suits in both of these pictures right here. So the reason behind this is that you can think about really tiny electronics, something as small as a piece of dust. If it lands on the electronic as you're building it, it's going to ruin it and it's not going to be usable, right? So actually, the suits that everybody's wearing, they're not to protect them from what they're making, it's to protect the device from us. So you can imagine your skin cells, your hair, your clothing, that all introduces a source of dust into these clean rooms. So it's a very, very well-regulated area, the temperature, the humidity, etc. So a lot of these technologies that I'm going to be talking to you about, they're actually the same ones that are typically used to make computer chips. So the things that go into your iPhones, any electronics. We basically repurpose those technologies so that we can make these tiny neural probes. So these clean rooms, they're given what's called classes. So ours in particular is called a class 100 clean room. So to give you an idea of what that means, it basically means in every cubic foot of air, there's fewer than 100 particles. So it's very, very well regulated, and that's how we're able to produce things at such tiny scales. So in terms of the basics of microfabrication, so how do we do this? So you can actually think about this as kind of building a cake layer by layer, and with each layer, we're going to lay down a different pattern. So what you start off with here is typically a silicon wafer, and you have a layer on top that you're going to pattern. You deposit what's called a photoresist. So this is basically a light-sensitive polymer that you put down and an even layer on top. What you then do is you shine UV light through a mask. So you can think of this as basically a piece of glass that has a pattern in it. And so as you pass UV light through, the parts that are clear, they're going to go through and interact with the photoresist underneath, whereas the darker parts, the light's just going to stop and not interact with that photoresist. So what happens then is you do what we call develop it. So think of it as developing a picture, for example. So basically you end up with the photoresist that did not interact with the light still on the surface. You can then etch this wafer. So basically you can put it in either a wet chemical bath, or you can do a dry plasma etch, and that'll etch away everywhere that's not protected by that photoresist layer. And so finally, when you remove this, you end up with the pattern that you'd like, and you just keep on moving from there until you build out your final device. So I'm going to talk about basically how this works specifically for making neural implants. So again, we start off with our silicon substrate. We're going to put down a polymer So that is what makes it flexible. We put down and pattern the electrode metal. We put down that polymer top layer, and then we etch through the polymer to define the area or the entire device, and then also down through and expose the electrodes. And so these metal traces, they lead all the way up to the top, where you can add electronics that allow you to communicate with the electrodes that you're trying to sense from. So again, this is made of polymer, so it's actually very flexible. So in order to actually get it into the brain, we attach a surgical insertion tool to make it stiff enough so that it can actually penetrate. So then what you do, it goes into the brain, you remove that stiffener, now your device is ready for sensing. So what's cool is that using a bunch of microfabrication techniques, you can basically make many shapes and sizes. These examples here kind of show how flexible these devices are. You can kind of bend and move them around, which is really great because that really prevents a lot of damage when you actually put it into the brain. So you can change not only the shape and size of the devices themselves, but also the electrodes. So basically we interact with a lot of different research institutions and universities with neuroscientists to see what they would like, and we try and engineer that to make sure that they can get the recordings that they want. So now I'm going to introduce our current device technology and capabilities. So what we're actually doing in the lab as engineers in order to get these devices into neuroscientists' hands. So the first step is, as I've mentioned before, the device fabrication. So you can see here, this is kind of like a cross-sectional view of what that might look like. So you have your silicon wafer here, the electrode metals, and then we actually put different layers of that polyimide polymer in between the metal to insulate it. So you can peel it off, and then basically your device looks like this. So next you actually have to add connectors, right? So you need to have something that people can plug into to make these measurements. So again, we talked to a bunch of different neuroscientists. There's different ways that you can add these connectors just based on what they would like. So the next thing we do is actually coat these electrodes with different coatings electrochemically in order to improve the ability for these electrodes to either sense or stimulate. So these are three examples of chemicals that you can actually use electrochemistry to put onto the device. So this one's called PDOT. The full chemical name is here. I'm not going to try and pronounce it and embarrass myself. But you can see in this particular case, these bottom row of electrodes, they're actually the bare platinum metal that comes out from the clean room. And then we deposit it selectively on these ones on top. You can have other chemicals like iridium oxide, so that's this light blue color. And what's cool is that you can really just cover the entire surface of whatever you'd like, regardless of the size in this case. And then you can also deposit metal alloys using electrochemistry. So this is platinum iridium. You can see the darker areas are that platinum iridium, and then the lighter areas are the bare platinum. And so finally, you have to prepare these devices to actually go into the brain. So what we actually do is use, again, microfabrication techniques, but this time on the actual insertion tool itself. So these are all made of silicon, and down this shank you have a thin little channel that you etch. So you can fill that with a temporary glue, such that you turn over the stiffener and attach it to the device. And now your device is basically ready for implantation. So here's an example of a device that's ready to go for surgery. You see the stiffeners right here. You have these wings here that help adhere the device to the skull, and then your connector up here. And this is just an example of looking through a microscope what it might look like with your device, and then the silicon stiffener kind of peeking out from behind it. So when you actually want to use these for surgeries, what you do is that you attach it to an arm, you insert it into the brain. You can then build up kind of a well around your insertion areas where you glue down those wings that I was showing before. You bend over the connector region, and so you can actually use saline and dissolve that temporary glue that I was talking about that connects the stiffener and the device, such that when you actually remove it, the device still stays in the brain. So this is what it actually looks like during surgery. So you have this arm that is attached to the stiffener. You see these two wings right here. And you can kind of see this ribbon that leads down. That's the device going into the brain, and then the silicon stiffener is right here. And then when you remove it, you see only the stiffener comes out and the device is still in the brain. So looking forward, ultimately, it's great to do these with animals, but we really want to be able to help people with our technologies, right? So at the lab, we also have the ability to make higher density recording and stimulation devices that can actually go into humans. So as Anna mentioned before, there's one of these technologies is called deep brain stimulation. So that's basically one of these probes. They insert it deep into the brain to address certain neurological diseases like depression, Parkinson's, etc. So commercially, there's typically 4 electrodes at the very end, but by using microfabrication techniques, we're able to pack in more over a longer length of this device, and we can put in 32 to 64 electrodes, which is great. We can also make devices that go on the surface of the brain. So this is actually one of our devices on a human brain. This was done at UCSF, and they're measuring electrical signals on the surface. And you can see also with these devices, you can really pack in a lot more using those clean room techniques that I've been talking about. So I've been talking about the devices, but we also focus on the electronics that are coming out. Because you might imagine, you need power in order to use these electrodes, and nobody wants to carry around a heavy backpack full of batteries and electronics in order to do this. So we're also looking at ways that we can miniaturize everything. So, this bar is about, I think it was, oh yeah, 1 centimeter, sorry, it's really hard to see. And then you can shrink it down to 1 millimeter right here. So everything we can shrink down, these are connectors, both of these. This is like your circuit board. So we can shrink everything down 100, 150 times to make everything smaller and more compact. So looking into actual applications, so one application of our devices is through an initiative called Brain Research through Advancing Innovative Neurotechnologies Initiatives, or the BRAIN Initiative. So this was started by Obama back in 2013. And the goals of this were to bring together both the public and the private sectors in an effort to both address neuropsychiatric and neurological diseases, looking at ways that we can understand and hopefully cure memory loss, looking again at peripheral nerves, so not only within the brain but in your spinal cord, and finally looking at large-scale brain manipulation technologies. So these are some of the funding agencies that are involved in this. And so we've been able to benefit from this initiative through a project funded by DARPA. So this is developing you can think of it as a brain pacemaker, ultimately for veterans. So with a lot of veterans, there's a lot of neurological diseases that they may be suffering from. For example, PTSD, brain injury, anxiety, depression. So there's actually a huge need to understand what's exactly going on with each of these diseases on a single neuron scale. So with our devices, we can kind of create a system where you can better understand in different areas of the brain what's going on with these sick people in a way that we can hopefully start stimulating the areas that might be different and hopefully help them. And so right now, these are kind of the state-of-the-art neuroprosthetics. Maybe you've seen some of these in the news. For example, this woman was able to control this arm and give herself a drink of water just by using her mind. This guy, or same with this guy right here. Again, we have the retinal implants. And then, maybe not as useful, but still cool, you can also, they've been able to use this brain interface to have this guy basically play a game with his mind. So that's been pretty cool to see kind of real life applications of how this technology has worked thus far. So to kind of wrap up this section, these are some of the concerns that we deal with as engineers when we develop these devices. So Anna and I have been talking about some of these before. So for example, looking at how you package the device, how you insert the device, integrating everything together, ways that they might fail, the difference between animal versus human use, looking at the electronics, making sure that everything is fabricated to the highest quality, and then making sure that everything is also biocompatible. So you might be wondering, I've shown you a bunch of cool things, Anna's showing you a bunch of cool videos, so what's next? What are we currently working on to kind of push that field forward? So next, Anna will be talking about some of the newer things that we've been working on.
Thank you, Allison. So I'm going to change it up a little bit. I felt kind of bad before when I kept turning away from you guys. So as Allison mentioned, We're looking at other new exciting things in the brain. The reason for that is your brain is more than just electrical activity. This is probably exciting to know because it's several pounds, right, that you're carrying around in your head instead of a small circuit board. And the reason for that is there's probably millions or billions of chemicals that are in your brain, in your whole body, right? Those are just as important as these electrical signals. They're what drive the electrical signals. And so figuring out, again, how to make these biomarkers kind of work is a key part. Examples of that. So again, dopamine. Have you guys heard about dopamine before? Serotonin, right? Crucial for things like addiction, in this case, schizophrenia and depression. Serotonin too, depression and anxiety. I don't think there's any person who's ever watched a single hour of television and not seen a commercial for an SSRI, which is again a serotonin reuptake inhibitor. Huge thing, right? Like we know this is important for brain function. Another one, acetylcholine, very important for aging, for Alzheimer's disease, memory in general. And then finally, GABA is another one that people talk about all the time. Oh, make sure you're getting enough of GABA. GABA is one of the big ones, along with glutamate, which I'll talk about in a second. So we're building ways to kind of interact with this chemical part of the way the brain communicates. And the way we're doing that is by building sensors that can directly look at the levels of different chemicals and integrating those onto these same electrical activity probes. We're also integrating microfluidic channels. So if you see both of the devices behind me have fluidic channels at the bottom. So that's a way to directly add drug to regions of the brain. So again, a lot of drugs have kind of symptoms that you see throughout your body. You can probably think of people, you might know someone who's been on a certain medication for a long time. It's starting to break down their liver in some way, causing issues with other organs, but they need to take it for the health of another part of their body. So the idea of this is, a local way to target drug delivery where it needs to be, or even in one brain region instead of throughout the entire brain. Another thing that we're doing, which we're not going to talk too much about today, but I encourage you all to get online and look at a lot, is integrating optical waveguides. So that's adding fiber optics and ways to put light into the brain. And the reason to do that is, if you guys have heard of CRISPR-Cas9, again, a group at Berkeley helped kind of figure out how this technology works, but it's a way to genetically modify cells to turn on or off their signaling using light. So integrating ways to actually get light deep into the brain is really critical to be able to use this new tool that biologists have developed. Again, because if you can use light to stimulate a specific cell instead of electricity to stimulate an entire population of cells, you've improved this therapy. So that's kind of the ideas that we're working towards. So I'm going to talk to you about chemical sensors because that's what I do as a chemist and that's what I'm excited about. So even after I gave the plug for optical waveguides, sorry. So chemical signaling in the brain is kind of the part that goes on between the electrical signaling. So again, you can see on the left, yeah, I got it right. On the left, that kind of larger picture showing the electrical signals. But if you zoom in, the space where neurons, kind of that tiny space between neurons is where this chemical signaling happens. And what happens is all of these electrical signals and firings that are happening are actually cells releasing chemicals. Those chemicals then will kind of go out and through these tiny little regions called the synapse, diffuse to the next cell where they hit receptors. And based on what happens at that receptor, based on that interaction, there's some kind of alteration that happens to the cell that then causes a signaling cascade that can either create that electrical activity that you typically see, which is what we're usually working with our devices, or it can create some kind of other effect. And so what we do is we kind of build, like, I guess, artificial receptors, or we create these biosensor surfaces. And what they are is the sensor surface interacts with the chemical the same way the receptors in the brain would interact with that chemical. And instead of starting a signaling cascade, what it does is we build these kind of special electronics that help amplify a small change that happens on that biosensor surface and turn that into something that we can read out on a computer as just a change. You can do this, that change can be like an optical light signal. It can be a frequency or voltage shift. Most of what I'm going to be talking about today are current changes that we'll see. Again, for those of you who do electronics things, this may not mean a lot. These are picoamp to nanoamp signals. They're very small. even when we amplify them. So what we're doing is looking for very, very small changes. And we're doing this in a very crowded, like chemically messy place. So imagine there's millions of chemicals in your brain, and we're looking for the binding of a few to a surface. So that's kind of what we specialize in trying to do and add to these devices. The chemical I'm going to use as an example for how these work, and a sensor that we've built so far, is for glutamate. So glutamate is kind of one of the big neurotransmitters in the brain. People talk about GABA and glutamate. Glutamate's kind of the transmitter that tells cells fire, start electrical activity, while GABA is the one that kind of, in general, ceases firing. That was a gross kind of oversimplification. If any of you are neuroscientists, I apologize, but that's the general gist of how they work. Dysregulation of glutamate, both the receptors that listen for that and the levels of glutamate in the brain are linked to a ton of different disorders. Autism spectrum disorders, migraine even, if any of you suffer from migraine, that could be a glutamate disorder, sorry. There's also addiction as well, all sorts of papers that have come out where understanding how this chemical changes or how to modify this chemical or glutamate in specific regions matters. It's also depression, but also specifically in certain disorders that have depression-induced, understanding glutamate levels and what happened during the trauma that in this case of this paper caused a stroke is key. So again, the way that we look at glutamate is using these sensor surfaces. So again, we have our electrical input, which is a surface, a signal that we put in, and then we're looking for changes in our electrical output. And that's because of that transducer element that's creating this change that we're looking at. And so what's actually happening on the biosensor surface is, Allison showed those slides earlier, right, where there was PDOT and iridium oxide and all these different coatings on the electrodes. So what we do is we take those platinum electrodes and we add a lot of special coatings to them. And it's a mix of selective filter coatings and coatings that interact with our chemical of interest. So in the case of glutamate, what we do is the outermost layer of our sensor is what we call our enzyme layer. So it is glutamate oxidase suspended immobilized on the surface. And essentially what happens is glutamate oxidase will only interact with glutamate. It's an excellent molecule in that sense. And what happens is when glutamate and glutamate oxidase interact, they create hydrogen peroxide. And hydrogen peroxide will then diffuse through this selective membrane. So again, right here, it just looks like a dark blue spot, but imagine this is a tiny microporous layer, sub-nanometer thick, or a few hundred nanometers thick. And essentially, you guys familiar with Plinko from The Price is Right at all, right? You drop a little token in and it kind of bounces around. The taller up that you have your surface, or if there's any charges to help knock something out. Essentially, a lot of things get knocked out or stuck as they try and go through kind of that Plinko chip. And so only certain molecules can make it through all the way to the bottom. And hydrogen peroxide for this particular coating is the only thing that can make it through. So then when the hydrogen peroxide makes it to this electrode surface, you get a change in the signal. So that's what you can see. It looks like at about 25 seconds, both of these signals show a little jump, and that's hydrogen peroxide making it to the sensor surface. And again, anything that can make it to the surface will show that signal. But that's what we do. A lot of our job, and me as a chemist, spends a lot of time making sure these coatings work really well. So again, I explained to you kind of how the sensor in theory works. This is what the data from them looks like. So we calibrate these sensors in a beaker. It's almost literally what it sounds like. The beakers actually look a little bit like shot glasses. We have a little stir bar in the middle and the bottom so that the liquid is stirring. And we will just use a pipette, if any of you guys have used a pipette, to add increasing injections of both the chemical of interest, in this case glutamate, and other chemicals to make sure that this is a selective electrode that's only responding to glutamate like we hoped. So again, as a reminder, on the far left, you see what those enzyme layers are like. The layer with the enzyme and then the protective layer, and that protective layer is helping to reject almost everything. And then what we also have is what I call a control sensor. So that's the kind of cartoon to the right. And so that's the exact same layer on the electrode, but minus the enzyme. So that's kind of what we call a control sensor, just to make sure that all of these coatings that we worked so hard to build are really working. And so what you do is our glutamate sensor trace is in blue, and the control sensor trace is in black. And so what you're seeing is each time there's a green arrow, that's an injection of 20 micromolar of glutamate. And then you see a step quickly happen. So that tells you the sensor is responding to glutamate. And as expected, the sensor is not responding or the sensor that does not have the glutamate oxidase in the layer is not responding. Then what happens is we can do an injection of ascorbic acid, and you see no response on either sensor, which is exactly what you would hope for. And then you do the same thing with dopamine, and you see no response. Ascorbic acid is vitamin C. That is everywhere in your brain. It helps. It's kind of the same reason you drink orange juice, to help keep you strong, keeps your brain, scavenges free radicals and things that are trying to cause damage. It's everywhere in your brain, and it's the thing that we work hardest to get past. It's very easy to see on chemical sensors. So that's always the big thing that you're worried about, is how do you eliminate ascorbic acid from these sensor arrays. And then finally, what we do is an injection of hydrogen peroxide. So again, you can see a hydrogen peroxide injection you see on both sensors. So that tells us that that control sensor we built with no enzyme in it is in fact working, and the reason we're seeing no signal on it isn't just because it's broken, but that in fact all of the enzyme layer, or all of the protective layers are in place. So once we get this kind of stairway or step of glutamate, what we can do is take that's a calibration for an individual electrode. But what we do is we take a bunch of these kind of steps or injection patterns, and we turn this into an overall sensor calibration. So this is what we use to tell people the performance of our sensors. This is how we know how much glutamate they're sensing in vivo. This is how we know if they'll even work for someone's application. And so again, you get this line. It shows you the current response you would expect for given concentrations of glutamate. And we can then do a linear regression over the first part to see kind of where the linear range that this sensor works in. Kind of the stats on how it works are on the right, but the big important thing to know is it works in the range that you would see in the brain, is kind of the takeaway here. So this is kind of the key part. This is a lot of what we do, is figuring out how to make these sensors work in vitro. The next step, though, is once you've built them, integrating them onto arrays that can be used in the brain. So again, this is a microelectrode array designed to have chemical sensors and electrical sensors on it. The top is that zoomed out picture that Allison was showing you before. Again, I'm not sure if in the earlier pictures we had like anything for scale, but just so you know, everything Allison was showing you too, the same size or smaller than what we're showing here. So that's a human hair compared to the device, to give you an idea kind of how small everything we're talking about making is it's all within that tiny tip. And so the actual tip has an array of sensors on it. The larger discs are 50 microns in diameter, and they're going to be turned into chemical sensors. And the smaller discs are 20 microns in diameter, and those end up being the electrical sensors. Again, just to give you a scale. So what's really cool about this system is this is a cartoon that makes it simplified, but if you're interested in electrical engineering or anything like that, the lab is an excellent place to spend a lot of time looking at wires. There's wires everywhere when you run these systems. So again, what we have is two completely separate recording systems that get plugged into, in this case, the two connectors, which you can see on the bottom right, the top of this animal's head. That pink is dental cement. It's not brain, dental cement, I promise. But you can actually see we physically just plug both of these connectors into a lot of wires and amplifiers that go to recording systems. And then what we can do is get out from each one of these sets of sensors just real time looking at electrical signals. So what you're seeing at the top, that electrical brain activity, those are single firing events for a cell that happen. That's the shape. We use this little shape to tell you that it's a cell firing. If you look at the time scale, these happen very, very rapidly. I could not identify this by eye. This is why we need computers. The bottom is glutamate activity. These changes are happening on a slower time scale, but they're what regulate that firing that you see. And so what's interesting is here what you can do is, again, this is in the brain of an animal 3 days after the animal's undergone surgery. What you can see at the top, those are what's called histograms. And so essentially they're bins showing how many times the cell has fired or how many times this action potential shape has occurred in a set period of time. I believe, I want to say they're like minute bins, but the idea being one cell's firing a lot more than the other. That's the takeaway on that. And then on the bottom, what you can see is there's the glutamate sensor that's showing all sorts of glutamate activity, but then there's also one of those control sensors to let us know if the sensor's working or if there's any kind of weird random noise. So you can see, unlike on the previous slide where the control sensor just had that dead flat line, this one has some noise artifacts. But this is really exciting because now this means we can do things like start to test drugs for migraines and see what the changes are. But there is one slight downside. And so what I showed you was 3 days after implantation. 10 days after implantation, your chemical signals start to look like this. And you'll notice there's not much of a difference between the two signals, are there? Which means that something's breaking down. And with that, I will transition to how Allison's dealing with the fact that our sensors break down.
That's so exciting. No, you're fine. So, we want to incorporate both of these sensing techniques onto one electrode. So what's really cool is with our electrical sensors, this graph above me kind of shows the lifetime of the electrical sensor itself. So this was actually implanted into a rat. This was done in collaboration with Lauren Frank's lab at UC San Francisco. And you can see that this y-axis is basically measuring the number of neurons per electrode that they were able to pick up signals from. And then the x-axis shows the number of days they were able to track this signal. And you can see in this particular case, this device lasted for 160 days. So that's really, really impressive. You're really able to look at long-term changes in the brain using this technology. But as Anna mentioned before, when you incorporate chemical sensors, specifically enzymatic sensors, this is the actual lifetime. So it's a really, really tiny fraction of the potential of this device to last for months, right? So some of the reasons that we think this might be happening, it could be the electrodes breaking down. There might be fouling of the surface, so basically the surface gets too dirty and you can't pick up any signal from it. It just might be less biocompatible once you do implant it into the brain. But what I'm looking at in particular is the stability of this enzyme on the sensor. So the enzyme could either denature, so it's a protein, so it could break apart in the brain, or the enzyme could just completely be dissolved and lift off from the surface, and then you don't have a working sensor anymore. So actually what's happening is, the brain is a very, very complex environment. So we really wanted to break it down and see what chemical components might be causing this enzyme to degrade at a faster rate. So the way we do this is that we immobilize the enzyme, and the way we do that is by binding it to another protein called bovine serum albumin, or BSA. So then we coat the electrodes, so that's going to be these guys, the red dots right here. And then what we do is we run that calibration curve that Anna was talking about, where you change the concentration of glutamate, and you see how that change in current correlates. With that, we can create this curve where you can track the concentration of glutamate versus the current, and that slope is going to be your sensitivity. And what I did is I basically placed different groups of sensors into different soak solutions. So these were very, very simple. We just wanted to really take it a huge step back and say, well, if we isolate it and soak it in only one different type of chemical at a time, is that going to cause the enzyme to degrade? So I did 4 soak solutions. So this is just a standard saline solution. I added glutamate, ascorbate, and then peroxide. So this graph is basically what it looks like in real time as I run one of these calibration curves. So you can see I'm measuring the current over time, and each of these stepwise functions shows an increase in glutamate. So from this graph, I can transform it into a calibration curve. So you can see I'm measuring the different concentrations of glutamate and how the current changes. And this goes from week 0 to week 5. And you might notice as you increase the time that it's soaking in these soak solutions, that slope is getting smaller and smaller. So it's getting flatter and flatter, and that's indicating some degeneration of this enzyme. And so this is kind of the result of all of these soak tests. So this y-axis shows the % change in sensitivity from week 0, so before we started soaking. And you might notice all of these numbers are negative, so that's that's not great, right? It's showing that sensitivity is going down in all of these cases. But what's interesting is you might notice that all the devices soaking in PBS, so that's these blue bars, they're degrading at a slower rate compared to everything else soaking in glutamate, ascorbate, or peroxide. So something about the presence of those chemicals is actually causing this enzyme to degrade. So now we know to a certain extent one of the things that's causing the lifetime to not be great. So I wanted to look at ways that we can stabilize the enzyme. So one way is by incorporating these hydrogel nanoparticles. So this was done in collaboration with a small startup in San Diego called Aqua Regenerative Therapies. And basically what they do there is they create these hydrogels. And so this is your hyaluronic acid particle. And they actually add, it's a lot of chemistry where they add different functional groups, basically to help create these particles of hydrogels and then have those hydrogels bind to the enzyme. So what's nice about this nanosil is that it's based on hydrogel, as I mentioned before. It's non-toxic, and it's a non-animal source. So you might remember before that our immobilization techniques are based on a protein called bovine serum albumin. So that's based a cow protein, right? So if we want to look into actually putting these into people's brains, we don't want to be relying on other animals' products in order to get this to work. So basically you can bind this to the enzyme and you can then immobilize that onto the electrode surface. And then we wanted to see how this performed compared to the standard immobilization technique. All right, so here, these are, again, these soak solutions. So in this particular case, I'm only looking at the saline soak and then those glutamate soak solutions. And I'm comparing both our standard immobilization method and then adding NanoSil to that layer. So what's really promising that we initially saw is that the sensitivity is much, much higher compared to the way we normally do it. So we thought, wow, this is awesome, this is great. But you might see, after one or 2 months, that sensitivity drops pretty dramatically. So you can see there's huge decreases right here. But we started looking at this data, and you might notice that the change right here, using our standard method, it really doesn't change that much in comparison to this huge drop, right? So what we're actually thinking is happening in this particular case, this drop that we're seeing is not actually because the enzyme's degrading. It's actually because we're not binding this nanosil enzyme complex correctly to the surface of the electrode. So this really motivated us to look at ways that we can figure out other ways that we can immobilize the enzyme to the device surface. So another huge reason why we wanted to look into different immobilization techniques are that we actually do all of this by hand. So we're hand painting these enzyme solutions under a microscope. It's as painful and terrible as it sounds. And so basically, Anna showed this picture before showing that we're trying to coat only one of these electrodes and not the other. But under a microscope, this is actually what we're seeing. So we're basically using this giant tool right here and hoping that as you kind of poke the device, it's only going to coat here and not here. So we were like, okay, we need to move away from this, right? So we actually went back to electrochemistry, and we're like, okay, well, can we use an electrochemicals technique to now coat this enzyme solution? So we looked at a really cool way we're doing this, where instead of using that BSA method, we're now looking at ways that we can use another polymer called chitosan. So the way this works is that you have your enzyme or your electrode array right here. You're going to pick one site to be your sensing site. The other site is going to be no enzyme on it. You can immerse that into a solution of chitosan. And so chitosan basically in really acidic solutions, it dissolves. So it's going to just be a basic liquid. But when you apply a potential to just one of these sites, so whatever site you want to deposit your enzyme on, that's going to cause the pH to change only around the electrode site. And so what that actually does is it makes it more basic. And that causes the chitosan to kind of salt out of solution and selectively coat only on the electrode. So we can then take that device out of the solution and put it into a different solution of enzyme now. So in this particular example, it's glucose oxidase to sense glucose. And what happens is the glucose oxidase will only bind to wherever it sees chitosan, so only really on those electrodes. So this is what we see after we do this chitosan method. And you can see wherever there's a blue arrow, that's where we were like, okay, we want to only deposit enzyme on this site. And you can see these bubbles right here kind of showing how selective we can get this So this bar right here is 50 microns. You're imagining, again, we're trying to deposit on these 50 micron size electrodes. So it's very, very comparable. So that's really great and promising. Additionally, what's been really nice is that the sensitivity of these sensors when we incorporate the chitosan is actually much, much higher compared to using our standard bovine serum albumin method. So in this example, we've been able to get sensitivities that are about 13 times better compared to our normal immobilization technique. So what we're currently working on next is improving the selectivity. So that's making sure that we can get this sensitivity on this control site as close to 0 as possible. All right, so to kind of wrap up this biosensor, so looking forward, these are kind of the things that we always worry about as engineers when we create these. So I was showing you that other one where we're looking at what engineers worry about when you're making the device. These are the things that we think about when we're actually making these biosensors. So for example, we talked about the selectivity, making sure that the device is sensitive enough to pick up what you were looking for, making sure that it actually lasts a long time, and then making sure that you can also integrate it properly so that you can do other sensing techniques in addition to the chemical sensing. So to kind of wrap this up, really, really far in the future, how could we integrate both of these technologies that Anna and I talked about today into kind of one device? You could think of, for example, a self-regulating medical device. So for example, say you have a chemical sensor on your spleen, and your spleen helps with your immune response. And the chemical sensor might pick up and say, this is not behaving the way I would expect it to be. So it could then send a signal to a neural device that could be implanted further up in your body, so your vagus nerve, and that could cause the device to say, okay, I got to stimulate this device or stimulate this nerve and bring this immune system back up. So that's really kind of what we're aiming for in kind of the far future. All right, so kind of in conclusion, it's not just me and Anna in the lab all the time. It's a huge team of people that have worked with us, a broad range of people. I get to interact with a bunch of different engineers, neuroscientists, biologists every day. We're always learning more. And these are our funding agencies, so the National Institutes of Health, the internally funded LDRD, and then DARPA. So at this time, we'd be happy to take any questions that you might have. Thank you.
Questions? I can do real quick. So that every like that.
So you were asking if this is the same kind of technology that could be used to control your phone or something like that, or again, control your computer? Yeah, so if you remember the video that Allison showed where a guy was wearing a cap and then moving his fingers to be able to control what was going on the computer, the idea would be a lot of the devices that people are using are just to better understand what's going on in the brain. And so once you get more information about how the brain works or which parts of the brain are saying different things, you could use them that way. There are speech prosthetics that are being developed right now, and with the speech prosthetics, what they're actually able to do is tell someone a sentence. That person can then think the sentence, and if they have an electrode array on their head, a computer can then pop up the sentence they're thinking. So again, people are working towards those kind of things, but they're still very limited. Honestly, for a lot of this, what's missing is the computational side. So if any of you are interested in computers, there's tons of work to be done in figuring out when you get all of these complicated signals from the brain, what's happening. But yeah, I mean, that's kind of one of the less maybe therapeutic ways that the instrument, or that people are trying to use this technology.
All right, watch your feet and fingers there. Where are the dice?
Thanks for your talk again. Can you talk a little bit more about the complications regarding biocompatibility and how is the body rejecting this? Have there been strides in better biocompatibility essentially?
Yeah, that's a really good question. So that's always a huge concern because basically anything that you're implanting into the brain knows it's not supposed to be there, right? So we always have to worry about some kind of immune response. So actually one thing that can happen is that you are causing, causing tissue damage as you put these into the brain. And the brain's going to start forming what's called a glial scar around the device. And basically over time what happens is that you lose any signal because you have this huge scar tissue around it. So there's a lot of ways you can address that. So that's actually why we use these polymer flexible arrays. So what's commercially available are typically based on silicon or some other harder material. And think about your brain as kind of like a jello. If you put a needle in there, your brain is not just completely stationary at all times. So as it moves around, it is going to cause more damage if you have like a hard needle in there. So with these polymer arrays, it's kind of moving with the brain. And so you're causing less of that damage, and we're looking at hopefully causing less of that glial scar.
Question. Anyone? We have one over here.
Hello. So I was curious because you said that you guys use like a silicon substrate, but from what I know, general from modern semiconductor manufacturing, usually just the pure silicon substrate isn't very common, particularly with MEMS or microelectromechanical stuff. But I wasn't entirely sure. So, why did you guys choose that?
So the silicon substrate, so you're talking about when we actually make the devices, right?
Yeah, particularly why you use a silicon substrate rather than some doped, you know.
Right, right. So I think a lot of it is maybe out of commercial convenience. So really you could have anything that's at a certain thickness and the width of the silicon that can go into all of these microfabrication processing tools. But everybody uses silicon as kind of like the basics for microfabrication. So these are very easily obtainable, these wafers, and we don't need to worry as much about the purity because they're really only kind of a vehicle to build the device on. So we just actually peel it off from the wafer, and then that's, we're not actually using the silicon.
I would say as a pure logistics, a regular silicon wafer is a lot cheaper than silicon oxide or some of the other ones. So if we're using it as a holding wafer, we pick the cheapest thing. So if it breaks, we're not as upset. Just like pure logistics.
Any other questions? There's one in the back there.
So first of all, thank you very much for presenting this wonderful talk. I hope you enjoyed the crowd. Would you share with students maybe a little bit about your path to where you got? Because I think we have a lot of aspiring scientists in this room.
I am originally from Austin, Texas. I was really into chemistry when I was in high school. I wasn't sure if I wanted to go into chemistry or neuroscience, and then I got a scholarship to go into chemistry, so I decided I wanted to be a chemist, just to be completely honest. But it worked out okay for me. I went to Baylor University, which is in Waco, Texas, and while I was there, I actually had the opportunity to do undergraduate research, looking for metabolites of different explosives in earthworms. It wasn't quite as exciting as it sounds. It's like a giant vat of like a Rubbermaid tub of dirt with earthworms in it, and you would add like little bits of different chemicals and then see where in the earthworm body the chemicals ended up. I did realize I liked chemistry though, so I decided to get my PhD in chemistry at the University of North Carolina at Chapel Hill. So again, I went from the center of the country to the far East Coast. I learned about basketball instead of football, and I learned how to build chemical sensors. What I did was build specifically dopamine sensors for use in the brain. We were building them to look at how dopamine changes in response to different cues in an environment for regular learning, like when you're learning to get a sugar pellet versus if you're learning to get cocaine. So basically how drug addiction changes people's feedback to different cues. And then I came to Livermore because what I had been making in graduate school were made of glass and carbon fibers, which are basically little hairs encased in glass. And I thought there has to be a better way to make chemical sensors. And so I ended up here to do my postdoc. And I've been here for about 4 years at Lawrence Livermore.
And for me, so I actually grew up in Irvine, so Southern California. For my undergrad, I went to Brown University on the East Coast. So I did chemical engineering there. I actually didn't think I was going to be an engineer at all when I first started. I was one of those people when applying to colleges, I didn't really know what I wanted to do. It was vaguely science maybe. So actually I applied undeclared to a lot of places. And I kind of stopped by an engineering class at Brown, and I was like, oh, this is pretty cool. Like you're building things to help people. And that's what kind of put me down this path. I did also undergrad research at Brown University. What's really great is I think a lot of professors are really open to having students come and learn, because really all I did was I was taking a class and I was like, this guy seems cool. He's doing bio-related stuff. I might be interested in some kind of bioengineering application. And after class, I just went up to him and said, hey, I want to start doing some research. Can I help? And he said, sure. So that's, I think that exposure was what helped me decide I wanted to do graduate school. So I was over the East Coast, no more snow. I came back and did my PhD at UCLA, where I started getting into more of these neural sensors. And that was really just talking to different professors and picking what sounded the coolest to me. There was really no deeper thought other than that. But I'm really glad at the path it's taken me. And so I've been at Lawrence Livermore for about 2 years now. So.
We have a few more minutes for a question or two. Any more? Well, thank you so much for coming over and giving us a slice of what you guys do at the lab there. I know there's a lot more to that, so let's thank them again. Our speakers, Dr. Belle and Dr. Yorita.